Hi, everyone, and welcome to After the Award, where we are going to talk to our former Isaac awardees about their experience post winning the Isaac grant. So, if we can just get started with you telling me your name, the institution that you're connected to, and the year that you won your grant.
Absolutely. So my name is Chelsie Armbruster. I am an associate professor now at the University of Buffalo in the Jacobs School of Medicine and Biomedical Sciences. And the grant would have been awarded in 2024, I believe.
Why did you apply for the Innovative Science Accelerator Program grant? What was the inspiration for you? Was it just something that you had never seen before? Just tell us a little bit about that experience.
Yeah, absolutely. So I had come across the funding announcements a couple of times and was really curious about the mechanism. And research-wise, it was just a really perfect fit for something my lab was trying to do. So my background is I had been studying catheter-associated urinary tract infections, especially in patients with long-term catheters. And we had built this really exciting longitudinal data set where we did weekly assessments of signs and symptoms and urine culture results in patients. And we were seeing this unusual trend where we had individuals who were highly colonized by pathogens, but were not developing infection. And then we also had a patient who had a very stable, persistent, high colonization by pathogens and did develop an infection, but there was no change in what had been present in their urine for several months leading up to that infection. So it kind of challenged that idea of a catheter-associated UTI arising from a new pathogen coming in and immediately causing a response. So when we saw this funding mechanism, we thought this was the perfect opportunity to go to some of our safe samples and try to take a deeper dive into what's going on in that microbial community. So we put together a proposal to actually do shotgun metagenomics to kind of optimize the process for this, for working with samples from catheterized patients, and then use that technology to kind of dig in and see, is there something that's being missed by standard urine culture that could actually be more informative for risk of developing infection? So it's something that, we needed funds to be able to do it. We really didn't have any other mechanism for starting this work. It was not quite in a place where we could go for something like an R21 or certainly not an R01. So very high risk, high reward, and I was just really excited when I saw this opportunity. It seemed like a perfect fit. So
How were you made aware about the award, if you don't mind me asking?
yeah, from emails and also online notifications. Yeah. Yep.
That's so great. I'm so glad that it seems like you already had such a strong foundation. You just essentially needed the funding for it. And so I'm so glad that those two things were able to meet for you. What progress has your innovative idea, made since receiving the award? Like, have you been able to publish? Has it been peer reviewed? What's been going on since then?
Yeah, absolutely. So we're about ready to submit the publication. We're actually going through just the final, dot all the I's, cross the T's with the co-authors. So I'm really excited that we're going to post it on bioRxiv or possibly medRxiv in the next week or two. But it's really been amazing what this has opened up for our research. So the award allowed us to do the sequencing for longitudinal weekly samples from 9 study participants. So we had this really great view of the dynamics of what's happening in these microbial communities. And, we actually pulled in a biostatistician on the award who was able to apply a really fantastic framework for working with longitudinal samples to take the microbiome data basically and align it with things like antibiotic exposures and catheter changes and signs and symptoms that are part of CAUTI diagnostic criteria. So it's something that's never been done before in this space. So we're really excited to get that publication out, and it's, what we've learned from this work is going to need to be tested in a larger patient cohort, but we think that it is actually going to give us some potential species that might be biomarkers of infection risk, as well as a potential way to view urine samples differently in this patient population. So maybe maybe the things aligning with infection, it's not just the presence of pathogens in urine, but maybe we need to be considering the relative abundance of those pathogens in the context of all the other microbes that are actually present but not detected by standard culture. Yeah.
This is amazing. I mean, the work that you all are doing, and just even finding this different pathway, and I guess seeing a gap, and honestly being able to fulfill it, I think is just absolutely incredible. And again, we are so happy that this award is helping push this project along, and you guys are making such great headway. So how soon do you see the publication coming out?
Yeah, great question. And I have to say too, I mean, we could not have done this work without this award. So I'm so thankful for this. I hope something like the Isaac Project comes back again, because this was transformative for my lab. It really was. So yeah, hopefully we'll have the manuscript submitted in the next 2 weeks. Like I said, it'll be available in a preprint. We're aiming pretty high with this, so it might take a bit to go through peer review, but I'm going to optimistically say it'll be in press in 5 months, but you never know.
And as soon as it is, we definitely would like to receive that so that we can put that, of course, out so that we can share that with our audience, because I'm sure they would be very excited to see this incredible work that you're doing. What was different about the Isaac grant from other grants that you've seen in your research tenure?
Yeah, great question. I mean, I think the thing that I probably appreciated the most is that in writing it, the goal was to really convey what is the gap, what is the significance of what we're trying to do, and why can't we get this funding someplace else, instead of getting really bogged down in the mechanistic details of how are we going to do it, right? So obviously you need to provide enough of that to have confidence from the reviewers that you can do what you say you're doing. But I think just that freedom of, we need to tackle this challenge, and here's why it's really important, and what tackling that is going to open up for the field. So I very much liked that, and the fact that it was such a kind of short format for writing it too. Yeah.
Amazing, amazing. And I just have a few more questions. This is such great information. I think you've shared just a beautiful perspective on your journey thus far, and of course, all of the great work that you and your lab are doing at this time. I just think it's amazing. Where do you see the future of this idea going? I know you talked about nearing the end to getting all of the work published, and even just being able to work with a bigger human population as far as the studies and all of that. But where else do you see this going, and do you have a goal in mind?
Yeah, absolutely. So, I mean, like you said, the next step is going to be, can we validate our hypotheses that were generated through this work? And can we validate that in a larger patient population? Does this hold up if we start looking at a more diverse patient population too? I think we need to focus on the individuals with long-term catheters right now, because it just sets up a very unique environment in the urinary tract. Ideally, long term, I would love to see if we can also translate anything that we're learning from this patient population to other populations, like those with intermittent catheters, or even just uncatheterized individuals. But that's the big thing, is we need to have a larger study, have more individuals where we can kind of monitor and apply our hypotheses from this, and see if we look at the context of the entire urine microbiota, in addition to just what's present on a urine culture, is that informative for risk stratifying individuals, right? Who is at higher risk of potentially progressing from colonization to infection, who might need a more aggressive intervention, and who should kind of, what patients should we avoid antibiotic exposures and things like that. So I think that would be huge. But the other piece of it too is that we're really hoping that what we're learning from this could kind of move towards a diagnostic support tool. So that would also be amazing to kind of help define, is this really an active infection or not? Again, based on that whole community context of what's happening in the bladder.
Thank you so much for sharing. This is probably not going to be a typical question you've heard, but is there any, like, do you have any personal connection to this study or to this work that you're doing by chance? Or is there anything that, like, was personally kind of driving you to do this?
Mhm. Yeah, a little bit. So, I mean, intellectually, I am just fascinated by how bacteria interact with each other and in communities rather than as single cells. So I think that's a lot of what drove my kind of career path. But specifically focusing on nursing home residents and individuals with long-term catheters, it partly comes from experiences actually of one of my grandmothers. It made a big impression on me when I was little. She ended up in kind of assisted living and then in a nursing home care for probably the last 20 years of her life, and just watching things that she experienced and seeing the reality of having health issues in some of those facilities, especially in cases where the caregivers are so overwhelmed and overworked all the time, and you end up with a really vulnerable population that doesn't always get the attention that they need. So I think that really drove me to want to have research that's having an impact in that patient population. Anything that I can do that even modestly improves their quality of life is something that I will consider a really meaningful contribution. Yeah.
Thank you so much for sharing that. I always want to kind of draw an eye to the people behind the work, the
Yep.
people and the stories behind the research and behind the numbers and the stats and all of the things, because I think it's important. I think it's important for our audience to know, and anybody who is actually unaware with the work that you all are doing. I think that just being able to put a face and a story behind it is so important. So I just thank you so much for sharing that. And my last question is, what advice would you give to incoming researchers in regard to applying for grants like Isaac or any other grants they go after, and also just finding their way in their scientific research journey?
Yeah, I think the biggest thing is don't give up. Don't lose heart, right? This is a challenging career, but it is so rewarding. And I know it's interesting times for seeking funding, but you have good ideas. Believe in yourself that you have your good ideas. Keep trying for the funding, but get feedback. Seek people in your field and ask their advice. Have them read your proposals, have them give you suggestions on how to better convey the significance of what you're trying to do so that you have that just better shot at getting through the review stages. But I mean, really the biggest thing is just don't give up. Don't lose confidence in your ideas or yourself.
And I think that's some of the best advice. I mean, essentially the endurance of it all. I mean, it's what you're going to be able to see the results from all the hard work that you put in. So I just thank you so much, Dr. Armbruster, for sharing everything with us and letting us know your journey thus far since receiving the grant. And we wish you the absolute best. And that concludes After the Award for today.
Thank you very much. This has been fantastic.
Thank you.